You guys hear me now?
Yes.
All right. When I plugged in my thumb drive, I wasn't able to speak. So let's talk about this question later because this has taken me several minutes to get back online. So if there are any questions, I'm going to ask you to hold off on them and we will discuss them after the lesson because that was a disaster. I plugged in my thumb drive and then my speaker didn't work. So I moved to the other computer. All right, let's talk about what we're going to do today. Are you guys seeing my screen? Nobody's commenting, but I don't think I've shared my screen yet.
We can see your screen.
All right, where's the syllabus? So today is February 8th, and we should be finishing chapter 5. There is a quiz on Wednesday, which is actually Tuesday at 8 p.m. to Wednesday at 8 p.m. that'll be covering labs 5 and 6 and chapter 4 and the end of chapter 3. You should note that I have quiz 1, A and B graded, and I've started grading quiz 2A. I don't remember if I got everyone graded, but I got started at least on questions 1, 2, and 3 graded. So you can take a look at that before you take the quiz. I should mention that I did get the 2 last labs graded, so they are up. And I think the last worksheet, the lecture worksheet, has also been graded. All right, let's move to chapter 5. And I'll have to find out why when I plugged in my thumb drive, the speaker stopped working. All right, last time we talked about the nutritional classification of organisms, and we showed this slide showing you that you can split all organisms by their energy source and then further classify them by where they get their carbon source. So when we're talking about energy production, nutrient molecules have energy associated with the electrons that form their bonds. And how the nutrient molecules provide energy for us is we break those bonds, releasing the energy for our energy needs. And in cells, especially our cells, excuse me, hopefully you didn't hear much of that. In cells, energy is obtained by catabolic reactions, and the energy that's stored in the bonds of our food molecules, that energy is released, and some of that energy can be used to make ATP. ATP is the energy carrier molecule of cells. ATP has a high energy bond, the third phosphate group, very high energy, and it's an unstable bond. And the cell can break that third phosphate group off to release the energy when the cell needs energy. So ATP is called the energy carrier of cells. Any question about any of that? Energy is produced in cells, especially our cells, mainly by oxidation reduction reactions. Oxidation reaction is a chemical reaction where the electron is lost by the molecule or atom which is being oxidized. So oxidation is the loss of electrons. Reduction is the gain of the electron. You always have oxidation and reduction paired, meaning one atom or molecule, when it loses an electron, another atom or molecule gains the electron. So we say A is oxidized because it's losing the electron, and then B is being reduced because it's gaining the electron. Oxidation reduction reactions is a big, complex scientific term, and they shortened it to redox. So oxidation reduction reactions is the same thing as a redox reaction. Any question about that? An example of an oxidation reduction reaction is when copper +1 binds with iron +3. The copper is oxidized, losing an electron, so it becomes copper +2. The iron gains an electron, so it becomes iron +2, meaning add an electron, you go from +3 to +2. An easy way to remember oxidation reduction reaction is when you say the mnemonic, Leo the lion goes grr. Leo, lose electrons, oxidation. Grr, gain electrons, reduction. Any question about that? In biological systems, oxidation reduction reactions involve usually more than the electron. When the electron moves from one molecule to another, hydrogen ions usually also are removed with the electrons. Now remember, a hydrogen ion is a proton. And you take the hydrogen ion plus the electron, and then you have the hydrogen atom. So in biological systems, when we have a molecule which is oxidized, losing the electron, the hydrogen ion tends to follow that electron. And then this molecule, which is reduced, gaining the electron, gains a hydrogen ion. And in biological systems, especially for making ATP, the burning of glucose, an organic molecule here, loses the electron, which moves to NAD+. NAD+ gains the electron, so it's reduced. But NAD+ gains a hydrogen from glucose, and so it becomes NADH. And we talked about this. NAD+ changing into NADH is the electron carrier, moving the electrons around in a cell. It turns out NAD+ also binds with the hydrogen and moves that hydrogen around in cells. So in biological system, hydrogen ions or protons are typically also gained with an electron. As I stated, the biological redox reactions are used in catabolism to extract energy from the nutrient molecules, meaning the food we eat. And then we burn this food, and some of that energy is saved and used to make ATP. And that is our catabolic reactions that make ATP. Highly reduced compounds, meaning compounds that contain many hydrogen atoms, are degraded in our cells or catabolized, and then they're converted into highly oxidized compounds. For example, glucose is catabolized, C6H12O6, and it is oxidized into CO2 and water. These are highly oxidized compounds that have less energy than glucose. Some of the energy released by glucose is trapped and stored to make ATP. So these highly reduced compounds, meaning molecules like glucose, contain a lot of energy in their chemical bonds. And that energy is potential energy, which can be used to make ATP. Aerobic respiration is the main reaction in our cells that make ATP. And in all organisms which catabolize glucose, it is the main reaction to make ATP. A summary reaction of aerobic respiration is shown here. We have glucose and oxygen gas, which is converted by cellular respiration to make ATP and carbon dioxide and water. Along the way, some of that energy in glucose is converted into heat. Whenever we have energy converted in one form to another, meaning the energy in glucose converted to the energy in ATP and the energy in carbon dioxide and water, that some of that energy is always lost as heat. That's just a law of thermodynamics. You should know that cellular respiration, shown here, is used interchangeably with aerobic respiration. Any question about that? So these are two terms you should know, and they mean the same thing. The burning of glucose or some other food molecule into carbon dioxide and water for the purpose of making ATP is aerobic respiration. The summary reaction, C6H12O6 plus 6 oxygen molecules plus 38 ADP, adenosine diphosphate, plus 38 inorganic phosphate, make 6 CO2 molecules, 6 water molecules, and 38 ATP molecules. Any question about any of that? All right. Energy production and the generation of ATP. Some energy from redox reactions is used by the cell to make ATP. ATP is generated by phosphorylation. Phosphorylation is just the addition of phosphate, and that's inorganic phosphate, PO4 with a -3 charge, is added to ADP. So we have ADP, adenosine diphosphate, 2 phosphate groups, plus energy, and then we add another phosphate group to make ATP. You should note that the third phosphate group and the second phosphate group of ATP is a high energy bond. And when the cell wants energy, it can break one of these high energy bonds to release that energy and then get some of that energy for the cell's energy needs. Cells have three mechanisms for making ATP. There's substrate level phosphorylation, and that's where the cell transfers phosphate directly from a phosphorylated compound like this molecule here, which has a phosphate group and then 3 carbons. And the phosphate is transferred directly to ADP. That's called substrate level phosphorylation. It actually happens in our cells in fermentation and in glycolysis. And glycolysis happens in most cells. So it happens in most cells. And then the phosphate from this molecule is transferred to ADP to make ATP, and then the 3 carbon molecule. The second way that cells can make ATP is by respiration. This can also be called oxidative phosphorylation, but I won't use that term. I will just call it respiration. You should realize there's more than one form of respiration. Respiration involves a sequential transfer of electrons from an electron donor cell to other intermediates, and then the electron is passed to the last molecule, which we call the final electron acceptor. In our cells, which engage in aerobic respiration, electrons are donated by glucose, and then they're accepted by these energy carriers like NADH, FADH2, and then all of these molecules in the electron transport chain, which we will talk about them later. And then the electron moves from one molecule to the next, 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 until the electron moves to the last molecule, which we call the final electron acceptor. Along the way, energy is released, and some of that energy is used to generate ATP by chemoosmosis. And we'll talk a little bit more about that later in this lesson. Any question about that? Now it's shown here, it's not a big point about this, but when the electron moves from one molecule to another, that we have a reduction, which is the pink arrow. And then that's followed by oxidation as this molecule loses the electron, which is then gained by the next molecule in the chain. The third way that cells can generate ATP is by photophosphorylation. Photophosphorylation is only found in photosynthetic cells. Light energy releases electrons from chlorophyll, and then that light energy is converted to chemical energy via an electron transport chain. And we'll talk about that at the very end of this lesson. Along the way, ATP and NADPH are generated. But the ATP and the NADPH as well are often used to make sugar. And so photosynthesis is said to make sugar. Any question about any of that? And here we see a picture of photosynthesis. That's why the boys are so muddy. Any question about any of that? That was a little bit of a joke there. Photosynthesis is happening in the picture, but in the green plants in the background. So let's return to the topic of energy production. Energy is released from molecules in controlled ways and is stored by a series of reactions. The energy that's released in our cells by glucose, when we burn glucose to make ATP, is released and controlled in a series of reactions. If the energy were to be released all at once, there would be so much energy released that, well, the cell would fry. Because if you take glucose and then burn it by taking a blowtorch to it, the energy in glucose would be released immediately, and glucose would be converted into CO2 and water. So much energy would be released that you couldn't stand anywhere near it because of all the energy being released. It'd be too hot, as well as the fact that it'd be blinding. In our cells, electrons from donor molecules are passed from one compound to another through a series of redox reactions. And that's to release the energy slowly in controlled ways. Many organisms use carbohydrate catabolism as their primary source for obtaining cellular energy. Our cells do that. Glucose is the most common energy source, but cells may use other molecules like carbohydrates, lipids, and proteins to make ATP. And we'll briefly talk about cells using other molecules near the end of this lesson. We'll talk about carbohydrate catabolism, where the cell uses glucose. And we'll talk about that in depth. To produce energy from glucose, microorganisms use two general processes. They can use respiration and they can use fermentation. And I can mention that our cells can also obtain energy from glucose by using respiration and by using fermentation. However, our cells can only perform aerobic respiration. Microorganisms can perform aerobic respiration as well as anaerobic respiration, depending on the cell and what form of respiration it has. Some of them, like E. coli, for example, can perform both aerobic respiration and fermentation. Why am I getting this advertisement? Let me try that link again. Oh, here, no, that's not it. It looks like this link is not working. Let me try another browser. Oh, I don't have it anymore. Well, shoot, why is that not going? Well, that's not the right link. It looks like I'm not going to be able to show you this little movie about aerobic respiration. Let me see if I can just go to YouTube and find a quick one. Now, hopefully this video will be okay because I've never seen this one. This is just one I'm picking up quickly.
Respiration is just breathing, right? Wrong. Respiration is the chemical process that supplies the body with energy for all other life processes: growth and repair of cells, muscle contraction, protein synthesis, sending nerve impulses, absorbing molecules, and active transport, to name just a few. It happens in all living cells, not just animal cells, but also plant cells. There are two types of respiration: aerobic and anaerobic respiration. Aerobic means with air, and so needs oxygen, whereas anaerobic respiration doesn't need oxygen. Let's start by looking at aerobic respiration. Aerobic respiration releases energy in cells by breaking down food substances whilst in the presence of oxygen. It is represented by this simplified equation. But don't forget the energy which is released. Glucose is broken down by oxygen to release the byproducts of carbon dioxide and water. Energy is released, which is then used to make a special energy molecule called ATP. ATP is how energy is stored for later use by the body. Aerobic respiration happens all the time in all cells, usually in the mitochondria. Animals get the oxygen needed from the air through their ventilation systems. Plants get their oxygen from the air through the stomata. Do you remember how photosynthesis works? Compare these two equations. You can hopefully see that aerobic respiration works in the opposite way to photosynthesis. Plants produce their food by photosynthesis and then through respiration release energy from it. But don't be confused. Whilst plants do release oxygen as a byproduct of photosynthesis, they don't necessarily take in this oxygen for respiration. They just take in any oxygen from the air. Plants respire throughout the day and night, as do animals, but they only photosynthesize during the day, when there is light available. So this is how aerobic, or with air, respiration works. You just need to remember that glucose is broken down in the presence of oxygen to release carbon dioxide, water, and energy. In the next video, we will have a look at anaerobic respiration and how it differs.
Okay, any questions? Come on. All right, this slide is showing you a overview of aerobic respiration. You'll note that it has three principal steps: glycolysis, the first step, the Krebs cycle, and the electron transport chain. So this would be glycolysis, the Krebs cycle, and then the third step, the electron transport chain. Essentially, electrons flow from glucose, and then the electrons end up at CO2, shown here, and water, shown here. So electrons are energy-rich molecule. They have lots of hydrogen on them, and carbon dioxide and water are energy-poor molecules. From aerobic respiration, we get a high ATP yield, and that's comparing aerobic respiration to fermentation, whereas fermentation kind of has two steps. It has glycolysis, which is the start of fermentation. There's no Krebs cycle, no electron transport chain, but instead in one or two reactions, the pyruvate from glycolysis are converted into the fermentation end product. And it depends on which fermentation reaction it is, what the end product will be. but it could be ethanol or lactate. We do gain ATP from both aerobic respiration and fermentation, but the energy yield of ATP in fermentation is much less than the ATP yield in aerobic respiration. Any questions about this overview? Another slide showing you an overview of aerobic respiration. This cell is showing you aerobic respiration, which is happening in a cell, which would be the purple square. Aerobic respiration starts with glycolysis, and that's happening in the cytoplasm of the cell. Glucose and glycolysis is converted to pyruvate. The pyruvate then enters the citric acid cycle, which in our cells happens in the matrix of the mitochondria. Along the way, there's some electrons released by glucose, which flow to the electron transport chain, which also happens in the mitochondria. The pyruvate enters the citric acid cycle. The citric acid cycle does generate some ATP. It does generate CO2. And then more electrons from the glucose are taken by NADH2 and FADH2 and taken to the electron transport chain. In our cells, the citric acid cycle and the electron transport chain happen inside the mitochondria. In bacterial cells and other microorganisms, which I guess isn't true for yeast, but for other prokaryotic organisms, aerobic respiration does not happen in the mitochondria because prokaryotes don't have mitochondria. Aerobic respiration happens in and along the cell wall. Any question about any of that? Actually, the cell membrane, I should say, not the cell wall, cell membrane. As I mentioned, aerobic respiration is used synonymously with cellular respiration. And cellular respiration isn't quite as accurate a term because cellular respiration just means respiration in cells. And cells do not always have to accomplish aerobic respiration. As I mentioned, some cells like E. coli can form both aerobic respiration and anaerobic respiration. And whenever we use the term cellular respiration, we mean aerobic respiration. Any question about any of that? All right, so in respiration, glycolysis starts the respiration. It does have a different name, but I don't use it, the Embden-Meyerhof pathway. In glycolysis, oxidation of glucose occurs, and glucose is converted into pyruvic acid. So glucose starts it, and then glucose is converted into pyruvic acid. Pyruvic acid can be used interchangeably with pyruvate. The two molecules are slightly different from each other. Pyruvic acid has the hydrogen, pyruvate has released the hydrogen, but cells tend to use pyruvate and pyruvic acid the same. So we will use them to mean the same thing, pyruvate and pyruvic acid. Glycolysis happens in most living cells. Glycolysis can occur in the presence of oxygen, but it doesn't need oxygen. So glycolysis can occur in the absence of oxygen. In glycolysis, we do produce ATP and NADH, but we do not produce very much. This is showing you glycolysis in about 10 steps. Glycolysis is converted into this intermediate, converted into that intermediate, converted into that intermediate. This molecule is broken in two, this molecule and that molecule. This molecule converts to this molecule. This, whoops, went too far. This molecule converts to that molecule, converts to that molecule, converts to that molecule, converts to that molecule, and then converts to the last molecule of glycolysis, that is pyruvic acid. Along the way, ATP is generated. We generate 2 here and 2 there, but I don't know why I went back 2. I was trying to go back 1. But looks like I went back 3. We do need ATP for this step and this step. So we're adding energy to glycolysis in the form of ATP. We're adding 2 ATP, but we get 4 ATP out of glycolysis. So we say the net gain of ATP from glycolysis is 2. So the net gain of glycolysis is 2 ATP, 2 pyruvates, which are also called pyruvic acid, and 2 NADH. Now, I should tell you that when I took chemistry as an undergraduate, not only did I have to know all of the intermediates in glycolysis, but I had to know all of the enzymes as well. That's not even shown in this chart. Now, because it was required of me to learn all these intermediates, I think it's fair to require me, you to learn it as well. What do you think of that?
No way.
Way too much.
Good, huh? For glycolysis, you don't need to know all of the individual steps. You should know that we start with glucose, we end up with pyruvic acid or pyruvate, and along the way you gain 2 ATP molecules. You also gain 2 NADH molecules, and I'll talk about them in a little bit. All right, so you need to know everything in red from glucose converting into pyruvic acid or pyruvate. You should know, which isn't on this box, that with glycolysis, cells typically start with glucose. That is the preferred molecule to undergo aerobic respiration. Any question about any of that? All right. So here we're seeing an overview of respiration again. You should mention that we've only talked about aerobic respiration, which uses oxygen. And oxygen is the final electron acceptor in aerobic respiration. But there are other forms of respiration that do not use oxygen. The final electron acceptor is a molecule that is not oxygen. It's some other molecule that's not oxygen. And we'll briefly talk about anaerobic respiration. In respiration, ATP is generated by oxidative phosphorylation, or I should say largely by oxidative phosphorylation. And oxidative phosphorylation is the next step of aerobic respiration. So aerobic respiration starts with glucose, and we do generate some ATP too, and we do generate some NADH also too. And then we move the pyruvic acid into the Krebs cycle, which does generate some ATP too, and it does generate some NADH and FADH too. These molecules, the NADH here and here, and the FADH2 carry the electrons from glycolysis in the Krebs cycle to the electron transport chain, the third part of aerobic respiration. And then in the electron transport chain, ATP is generated by oxidative phosphorylation. And we'll talk a little bit more about that when we talk about the electron transport chain. All right, so after glycolysis, we enter the preparatory step. In the preparatory step, the pyruvic acid is prepared for entry into the Krebs cycle, meaning pyruvic acid cannot directly enter the Krebs cycle. It has to be oxidized and decarboxylated. And coenzyme A combines with the modified pyruvic acid, and that makes acetyl coenzyme A. Pyruvic acid is a 3-carbon molecule, 1, 2, 3. Acetyl coenzyme A is a 2-carbon molecule. So the first carbon of glucose is oxidized in the preparatory step, and we make CO2 in the preparatory step, as well as acetyl coenzyme A. Any question about any of that? All right. So for each glucose, we make 2 pyruvic acid. So that means the preparatory step runs 2 times for each molecule of glucose. Any question about that? I've already talked about this. I thought I moved forward. Here we go. All right. So the acetyl coenzyme A then can join with the Krebs cycle. You should know the Krebs cycle has other names. It's called the Krebs cycle after Dr. Krebs, who discovered it, but it's also called the tricarboxylic acid cycle because it involves many tricarboxylic acids. And it's also called the citric acid cycle because citric acid is the first molecule made in the Krebs cycle. And both the citric acid cycle are abbreviated TCA, and the tricarboxylic acid cycle is also abbreviated TCA. In the preparatory step, which is shown right here, pyruvic acid is converted into acetyl coenzyme A. The first CO2 is given off of glucose, and then the acetyl coenzyme A joins with the oxaloacetic acid of the Krebs cycle. The acetyl coenzyme A and oxaloacetic acid then make citric acid, which we're not going to talk about these, and then it makes this molecule, which then 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 makes oxaloacetic acid. And this is the molecule we started with in the Krebs cycle that bound to the acetyl coenzyme A. And that is why this is called a cycle. The Krebs cycle endlessly reloops, because essentially once you use up oxaloacetic acid, you regenerate it in the Krebs cycle. In the Krebs cycle, we make one ATP molecule, and that's shown right here, this step. We do make FADH or 4-NADH2. Excuse me, we do make 4-NADH plus. No, it's not a plus if it's NADH. Sorry, it's NAD plus, which is then adding on the hydrogen and the electrons to make NADH. And we make the first NADH in the preparatory step. We make one NADH here, one NADH here, and then the fourth NADH right here, right before we make oxaloacetic acid. Along the way, we do generate FADH2, and that happens here. And all of the carbon in pyruvic acid is converted into CO2. So the first carbon is converted into CO2 here. The second carbon is converted into CO2 here. And then the third and final carbon, oops, went too far, is converted into CO2 at this step. And that accounts for all of the glucose, or the carbons in pyruvate. It also accounts for all of the carbons in glucose. Glucose has 6 carbons. We split that into 2 pyruvic acid. Each of them have 3 carbons. And then the preparatory step and the Krebs cycle run 2 times per molecule of glucose. And so when we said that the Krebs cycle makes 1 ATP, you should realize the Krebs cycle does run twice. So the total ATP made by the Krebs cycle is 2. The total NADH made is 8, because we make 4 NADH in one cycle. And then we make 2 FADH2 with the preparatory step from the Krebs cycle and 6 CO2s. This accounts for all of the carbons in glucose are converted into CO2. NADH and FADH2 carry the electrons, which ultimately came from glucose, and they carry the electrons to the electron transport chain. All right, any questions about the Krebs cycle? So this is showing you the overview again. After the Krebs cycle, we then engage in the electron transport chain. In the electron transport chain, it's a series of carrier molecules which are in turn reduced and oxidized as electrons flow from one molecule to another. The electrons start from glucose and they're passed down this chain. That's why we call it an electron transport chain. And the electrons transfer energy from their molecule to high energy bonds, meaning the electron uses the energy in glucose to make ATP. And we'll talk about that a little bit in a little bit in more detail. Any question about any of that? You know, this browser isn't working. See if I can grab this.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Okay, any questions? I'm trying to see where they got things named. You do not need to know the names of the molecules which carry the hydrogen. Not carry the hydrogen, carry the electrons. You should know that some molecules in the electron transport chain. When the electron moves through the molecule gains the energy it needs to pump hydrogen ions from this side of the gradient to that side of the gradient. This was a pretty good video, actually. I think this is the one that my link was supposed to go to, but who knows? Yeah, it was, okay. All right. The electron transport chain in eukaryotes occurs along the inner mitochondrial membrane. In prokaryotes, the electron transport chain occurs along the cell membrane. So this membrane here has the molecules which engage in electron transport chain. NADH comes up to this molecule and donates the electrons, 2 electrons, to this first molecule. That gives this molecule the energy it needs to pump hydrogen ions from this side of the gradient to that side of the gradient. And then the electrons move to the next molecule in the chain. And then the electrons move to the next molecule in the chain, to the next molecule in the chain, to the next molecule in the chain. And then the electrons move from the last molecule in the chain of molecules in the membrane to the final electron acceptor. The final electron acceptor in aerobic respiration is oxygen. The electrons flow to oxygen. The oxygen combines with 2 hydrogen ions to form water. Along the way, hydrogen ions are pumped from this side of the membrane to that side of the membrane. This creates a twofold gradient, meaning there's more hydrogen, more positive charges on this side of the membrane than on that side of the membrane. So this membrane is negative compared to this side being positive. That means that the positive charges will want to be attracted to the negative charge. It also increases the hydrogen ion concentration outside the membrane. And hydrogen ions want to flow from areas of high concentration to areas of low concentration. This sets up a potential energy difference, and that potential energy difference is used by the cell to generate ATP. As the hydrogen ions move from this side of the membrane to that side of the membrane, they can only do that through this protein channel molecule, which is called, I just drew a blank on what it's called now. This is called ATP synthase protein. And when the hydrogen ion moves through this protein, it gives the protein the energy it needs to change ADP and phosphate into ATP. Any question about any of that? Here's a question for you. Is all of the oxygen you breathe in converted into water? Or is some of the oxygen converted into CO2?
CO2.
Anyone else?
We're talking about how some of the carbons get carboxylated, right, from glucose.
We're talking about how the carbons in glucose get burnt and converted into CO2. And that I'm not showing in this slide because that happened in glycolysis, or excuse me, not in glycolysis. That happened in the preparatory step and the Krebs cycle. So that's already happened. This is the electron transport chain where the NADH and FADH2 are donating the electrons into a chain of proteins in the membrane. And that's why it's called the electron transport chain. The electrons transport from one member protein in the chain to the next electron transport chain. So it's a chain of proteins in a membrane. And as the electrons move through some of the proteins, they get the energy they need to make the hydrogen ions move from this side of the membrane to that side of the membrane. That creates a twofold energy gradient, which is used to make ATP. Okay, and my question was, is all the oxygen you breathe in converted into water? And the answer is yes. That's why you must breathe in oxygen. And all of it is converted into water. None of that oxygen is converted into CO2. The CO2 that you breathe out comes from the burning of glucose. Any question about any of that? Let me shrink that down so I get all of it here. In the electron transport chain, you should know that ATP molecules are made at a high rate in the electron transport chain of aerobic respiration. In eukaryotes, they generate 32 to 34 ATP for each molecule of glucose going through the electron transport chain. Prokaryotes are a little more efficient. They generate 34 molecules of ATP. Now, in our cells, the cells which can generate 34 are the heart, the liver, and the kidney. And those are the only cells that we have that generate 34 molecules of ATP in the electron transport chain. All the other cells in your body only generate 32 molecules of ATP per the electron transport chain. And the heart, the liver, and the kidney are more efficient because they're constantly working. They're constantly burning or using up ATP for their energy needs. So these cells are a little more efficient than the rest of our cells. And I think you all know your heart has to continuously beat until the day you die. And so the heart is constantly beating. Okay, and it's true with the liver and the kidney. They're constantly working. They never get a break. All the other cells in your body get a break all the time. The final electron acceptor in aerobic respiration is a molecule of oxygen. It combines with hydrogen to form water. And that's why we have to breathe in oxygen to form the water in the electron transport chain of aerobic respiration. Any question about any of that? Some of that water we breathe out. Some of the water is used by the body for the body's water needs. Desert animals are very good at converting oxygen into water and aerobic respiration, and then using the majority of that water for the animal's water needs. But we don't live typically in the desert, and so we're not as good at converting or saving that water for the body's water needs. All right, this is showing you the electron transport chain and chemiosmosis in an overview. And this part of the electron transport chain is where the electrons move from one protein in the chain of proteins in the membrane, but the electron eventually flows to oxygen in aerobic respiration. And then this is showing you the electron transport chain, how the electrons are given to the proteins and then move through the proteins in the train of proteins in the membrane, giving some of the proteins the energy they need to pump hydrogen ions from this side of the membrane to that side. And then those hydrogen ions are used to make ATP. And this slide is showing you that in eukaryote cells, the electron transport chain happens in and across the inner mitochondrial membrane. But in prokaryotic cells, the electron transport chain happens in and across the cell membrane. In prokaryotes, where are these hydrogen ions pumped? It's outside the cell membrane, but where are they pumped? Nobody has a guess? These hydrogen ions are pumped in the cell wall. I already talked about that. I talked about that.
Are you able to zoom out a little bit to see the whole slide?
The only thing I need to talk about here is the electron transport chain is sometimes called oxidative phosphorylation, which isn't there, but it's right here in chemoosmosis. I don't use those terms. I just call it the electron transport chain. I think your book talks about the term chemoosmosis. Chemoosmosis is the pumping of the hydrogen ions and then the moving of the hydrogen ions to make ATP. And that's also called oxidative phosphorylation. That is a term that I don't tend to use. I tend to call it the electron transport chain. I should warn you that that's also called the electron transfer chain and the electron transport phosphorylation or the electron transfer phosphorylation. All of these ETC words are similar, so it doesn't confuse students. But chemoosmosis and oxidative phosphorylation are different terms that mean the same thing, but I don't use them just to not confuse you. So in aerobic respiration, ATP is made, and in eukaryotes, we get a total of 36 to 38 ATPs from aerobic respiration. And I'll show you the overview in a little bit, but two from glycolysis, two from the preparatory step of the Krebs cycle, and then 32 to 34 from the electron transport chain. In aerobic respiration of prokaryotes, they're a little more efficient. We generate 38 ATP molecules. And here's the overall summary of aerobic respiration, which you must know. C6H12O6 glucose plus 6 CO2 generate 38 ADP, and plus 38 ADP and 38 inorganic phosphate generate 6 CO2, 6 water molecules, and 38 ATP. And this is showing you what I told you, 2 ATP in glycolysis, 2 ATP in the Krebs cycle, and then 32 to 34 in the electron transport chain, and prokaryotes always generate 34. And as I mentioned, anaerobic respiration uses oxygen. Aerobic respiration uses oxygen as the final electron acceptor. Anaerobic respiration uses another molecule other than oxygen. And it can be many different molecules, such as nitrate or nitrite, sulfate. Generally, anaerobic respiration has glycolysis in the Krebs cycle, but it has a shortened electron transport chain. And this shortened electron transport chain yields less ATP than aerobic respiration. It depends on the form of anaerobic respiration, how much ATP is generated. But generally, it's someplace between 2 ATP, the very worst, and then the very best of anaerobic respiration generates 36 ATP per molecule of glucose. Because anaerobes generate less ATP than aerobes, anaerobes tend to grow a little slower than aerobes. Any question about this slide? If not, I'm going to stop here and break for the lab. We're a little bit behind schedule, but we will continue from here next time. All right, any questions? If not, I'll see you in the lab.